All right, so this is going to be a recording for looking at having us draw out all of the steps of photosynthesis. We're going to do an overview, we're going to do a look at the light-dependent reactions, and we're going to look at the light-independent reactions. So I'm going to go ahead and share my screen so you can see my drawings on the iPad. And, oops, come on. Okay. So, okay, so we are going to do, I'm going to start with, so just like you did a pamphlet for your cellular respiration, you're going to be responsible for drawing one out for photosynthesis as well, right? So I'm going to start with just the overview. So this is going to be, oh my God, my husband's in the background making a ruckus. A ruckus and muckus. There he is. There he is, making a ruckus. So this is going to be an overview of photosynthesis. And he is making lunch because us heterotrophs have to eat our food, whereas plants can make their own via photosynthesis. All right, so the overall equation, we're going to have 6 CO2 plus 6 waters plus the energy from sunlight. And that is going to be converted into glucose and 6 oxygens as waste, all right? If you look at this reaction, let's look at what is oxidized and what is reduced. So there's a big misconception that the carbon dioxide turns into oxygen gas, and that is not the case. Always follow the carbons. So here, our carbon dioxide is actually going to be converted into our glucose, right? And we're adding hydrogens, so that is going to be a reduction, right? If we look at the other, this water is going to turn into oxygen gas. So water is going to be split in the process of photosynthesis, giving off oxygen, and that, because we are removing an H, remember I always put an X through it, so that is oxidation, right? So these are redox reactions, oxidation and reductions, all right? And the overview of photosynthesis, there's kind of 2 processes. We have the light, oh my gosh, dependent reactions. Oh my gosh, Sophie, we need to zip it. And the light oh my God, he's still going. And the light independent reactions. All right, so let's just write about those first before we kind of draw out the overview. So the light, let me get my notes. I don't have them here. What else? We can just do it. So the light dependent reactions are going to be, they obviously need sunlight, right? Hence light dependent. They're going to occur in the thylakoid membranes, right? They're going to occur in the thylakoid membranes. We are going to see water split, and when water breaks down, you're going to get 2 protons, 1 oxygen atom, so half an O2, and you are going to also get 2 electrons, right? So those electrons are going to be really important for the outputs, right? So what you're going to get out is that you're going to get oxygen gas out, and we are also going to see that you're going to get ATP and the production of NADPH, right? And that is what is going to help drive the light independent reactions, right? So in the light independent reactions, we have that no sunlight needed, right? No sunlight is necessary. These are going to occur, this is going to occur in the stroma. It is otherwise called the Kelvin cycle. And I always remember that Kelvin in the cycle reminds me of carbons. We're going to be dealing with carbons, right? So in the light dependent reaction, we're going to use ATP and NADPH from the light dependent reactions to create C6H12 right? So if we look at, for example, a chloroplast, what we're going to see is that we have these thylakoid membranes. Remember, one full stack of pancakes is called a granum. And we're going to see water is going in to the light-dependent reactions, right? We're going to see electrons being shunted around, but two electron transport chains, we'll get that when you draw that out. But when water splits, you're going to get electrons, you're going to get some hydrogens, and you're going to get oxygen. That oxygen is going to leave as a gas, as a waste product, and we're going to see sunlight that is going to go in and hit those electrons to energize them, to move them around so that we can generate ATP and NADPH, right? In the light independent reactions, we're going to see that carbon dioxide is going to come in to the stroma. We have a cycle, which is called the Kelvin cycle, right? We're going to see that carbon dioxide is converted into glucose, right? And in the process, it's going to use that ATP, and it's going to recycle out the byproducts of that, which are necessary for the light-dependent reactions. All right, so that's the basic overview. Now we're going to go through each reaction separately. So this is going to be the light-dependent reactions. All right, so these are going to be the light-dependent reactions. I'm going to do a very brief overview, and then we're going to get kind of more microscopic to look where these events are happening. All right, so we are going to see that we have sunlight coming in. It is going to be hitting photosystem 2, right? And photosystems are just proteins and pigments, and they're on the thylakoid membranes, right? At the same time, in the thylakoids, we're going to see water split. Water gets broken down into two hydrogens, or two protons rather, and we're going to get some oxygen. The electrons that are given off, I'm going to make that a little bit bigger. The electrons that are going to get given off, these two electrons, are going to be energized by that photon of light, right? So these electrons are going to get energized, and they're going to go bounce, bounce, bounce. I don't know how many bounces they go on, but they're going to be going down ETC2, right? Electron transport chain 2. In the process of that, they are going to pump hydrogens into the thylakoid membrane. Those are going to be able to flow down their concentration gradient, and that is going to result in the production of ATP, right? So those are going to result in the production of ATP. All right, so however, we're not done. So those electrons are going to continue moving, right? And they're going to continue moving into PS1, photosystem 1. And those are again, so these electrons keep moving. This is again energized by a photon of light. And that is going to cause those electrons to then move down ETC1, right? So these electrons are now going to keep bouncing and keep bouncing, and those two electrons are going to finally join up with our friend NADP+, and that is going to create NADPH. right? So the two main outputs are ATP and NADPH. All right, so let's draw how this looks on an actual thylakoid membrane. And I don't actually think I've actually drawn this out necessarily. So we'll do that together. Okay, so we are going to be drawing, I'm going to basically draw one big thylakoid membrane here. Remember, there's going to be other ones that are kind of stacked in this pancake as well, right? So remember, out here is the stroma. Inside are going to be the thylakoid, or the thylakoids, right? And the membrane, the thylakoid membrane, are here shown in black. Ow! Just stubbed my toe. Ow! Toe stubber. All right. So we are going, I'm going to draw some things on this thylakoid membrane. First thing I'm going to draw is, whoa, that was a big old eraser. I'm going to draw photosystem 1, and I'm going to draw photosystem 2, and I'm going to draw a little space there. All right, so we have PS2, right? And we know that we're going to also have PS1 on these thylakoid membranes, right? The other thing that we know we have on the thylakoid membrane is that we have this kind of lollipop shape ATP synthase. And we also know that we have some proteins along the thylakoid membrane, right? And those are going to be represented. This is going to be ETC2, and this is going to be ETC1, electron transport chains. All right, so in the thylakoid membranes, what we're going to see is that water gets split, right? And when it gets split, we're going to see oxygen. We're going to see some oxygen. We're going to see some protons. Notice that the protons are already starting to build up in the thylakoid space, the space inside the thylakoid membrane. And then we're going to get, and I'm going to draw the electrons in red here, 2 electrons, right? Those 2 electrons are going to go into PS2. The oxygen is going to leave as waste. It's going to go out the stomata. But these electrons are going to get energized by a photon of light, right? When those electrons get energized by a photon of light, they are going to go through the electron transport chain. As they do that, we are going to see that H's get pumped into the thylakoid space. Not the stroma, but inside the thylakoid membranes. Those H's will be able to flow down their concentration gradient into the stroma. And when they do that, we are going to see the generation of ATP. Right, so here's our first ATP. All right, and that obviously comes from ADP and PI. Okay, these electrons, however, don't stop there. They keep going. So now they're going to enter PS1. And we're going to see another photon of light. And these electrons will go through ETC1, right? Those electrons in ETC1, I'm going to kind of draw it up here. The electrons in ETC1 are going to join with the final electron acceptor, which is going to be NADP+, to create NADPH. Also in the stroma. All right. So that's kind of photosystem 1, where we have PS2, electron transport chain, creates an electrochemical gradient that will be used to drive ATP synthase and the production of ATP. Those electrons keep moving to PSI1, where we are going to get that they go down the electron transport chain 1, and they're going to join with their final electron acceptor, which is NADP+, to create NADPH. All right, so that's the light-dependent reactions. Now we're going to draw out the light-independent reactions. All right, so the light-independent reactions occur in 3 steps. All right, and again, the light-dependent reactions are called the Kelvin cycle, right? That's another word for the light-independent reactions. And we know this happens in the stroma. So the first phase of the light-independent reactions is called carbon fixation, right? And that is where we get carbon dioxide out of the air, and we're going to put it into an organic molecule that the cell can actually use. We're then going to see the synthesis of G3P, which is a 3-carbon molecule that is going to be used to drive the production of glucose. And then the third one is that we're going to get regeneration of RUBP, which is a 5-carbon molecule that kind of starts the process again. Oh my goodness, there's a lot of motion in here today. Okay, so what we're going to see, and this is again happening in the stroma, right? And remember, from the light-dependent reactions, we got 2 things. Oh goodness, guys, please be quiet. We have ATP and NADP+. Those are from the light-dependent reactions. All right? So what we have for the light-independent reactions is we're going to have carbon dioxide coming into the system, right? Remember, carbon dioxide is a 1-carbon molecule. It is going to join with our friend RUBP, which is a 5-carbon molecule, right? 2, 3, 4, 5. I always draw them as little circles. And we have carbon dioxide, which is 1, right? As carbon dioxide enters, that is going to get added to RUBP, and that is going to create 1, 2, 3, 4, 5, a 6-carbon molecule, which quickly turns into 2 3-carbon molecules, right? You don't have to know the name of that necessarily. It's G3, or no, it's whatever. You don't have to know it. And then in this cycle, one of these 3-carbon molecules is going to go out of the system, right? While another one of the 3-carbon molecules is going to get shunted on to regenerate RUBP, all right? So if we kind of color coded here, we're going to do carbon fixation here in blue. That would be the carbon dioxide coming in, joining with RUBP to create our organic molecule, all right? Now, if we do synthesis of G3P in purple, that is going to be here where we are going to, actually, hold on. That is going to be down here when we make the G3P, where the 3-carbon molecule, which is going to be used to make glucose, right? This 3-carbon molecule is going to come out of the system and join with another one that had come out of the system before, and that is going to make our C6H12O6 glucose, right? Now, the regeneration of RUBP is going to happen here, where we start regenerating and we have to remake our RUBP in order for the cycle to continue. Okay, so ATP and NADPH are used throughout this process. You don't have to know exactly where, but you do have to know that this takes a lot of energy to fix carbon and then kind of convert it and create glucose, right? And in this process, as we do it, we are going to get a waste product of ADP and NADP+, which are both required for the light-dependent reaction, right? So we have these kind of, in this Kelvin cycle, we have now converted carbon dioxide gas into a sugar, which is pretty cool. Okay, I think that concludes the drawings for photosynthesis. Chip, chip, cheerio. Toodles, poodles. Adios, amigos. Bye. Bye, birdies. Oh, I'm losing my mind.